we have a few moments for a question or a comment from the board? Joan.
Question. I take the work you've done. Thank you so much, Claire. This is wonderful. I have a mother who died with diabetes, and she had a difficulty in sores that wouldn't heal, and so I'm seeing it, perhaps a relationship scientifically. I don't know if there is or not, but I'm just curious about that, and also would like to have sort of a sentence in plain English that would help me convey what is going on with this, because it sounds like obviously there's been some wonderful progress, but that there's a wider field that is not served yet, and I'd like to understand where we are, and then ultimately, would more money speed that up?
So I'm going to have Dr. Leach and Dr. Lee come up here and answer that question, and while they're coming up here, I will just say that we and other teams are also working on creating basically artificial skin in this same concept of healing in defects. So different tissues, but the same concept of growing replacement tissues. And so the question is, would more money help speed this up? And where would you go?
And on behalf of the fans of the San Francisco Giants, this bone repair issue is of great importance right now.
You're absolutely right. Thankfully, Buster is behind the dish again, and we're actually looking forward to seeing him this weekend down in Arizona. But as far as to answer a one-sentence question, at least in my research and the research that many others are doing, we're actually actively using FDA-approved materials in our small animal models to help streamline this process and get it to human patients faster. At UC Davis, we also have an outstanding center for Clinical Translational Science Center, which is there to help us design clinical trials and move these materials forward. But you're absolutely right. More money from the basic science side would help tremendously, because that allows us to investigate the full span very rapidly on multiple animal models and for different clinical indications.
Yeah, and just for me, you know, the science is great, the collaboration is beautiful, but for us, the scaffold technology, specifically the carrier from the clinician's perspective, is really the limitation. We know the cells and the potential of the cells, and it's important to understand how they interact with these scaffolds, but for us, working with Kent and having the ability to prove the concept, and with more money, obviously, we could prove that concept, because I think we're so close to the clinical application. I think the Kent's use of the FDA-approved materials has been critical, but we do have some questions that we want to answer right before we get to those human trials using the composite scaffold cell technology, and that's what we need the money for.
Dr. Lubin.
So that was a great presentation. I was just curious, being in a trauma center where you see a lot of these kinds of things, at what time, when do you consider you're going to intervene with this therapy in addition to what you do standard, or would you try standard, and if it doesn't work, then go to this? Because that's going to be an important question, obviously.
So in terms of intervention, we see a little bit of a skewed population, because we're sort of sent the worst of the worst. So we see a lot of patients who've come to us after failure. So I guess for us, the way we looked at our clinical trial and the way that it was designed really was as an alternative. So at the beginning. So traditional techniques are there. We know that we can form bone with iliac crest graft and with recombinant proteins on commercially available scaffolds. We just don't feel that they're reliable for larger defects or recalcitrant problems. And so because many of these patients have actually had many of these things done to them already, it's an option for the patient from the beginning.
And do you think in congenital defects where there's a discrepancy in leg length, you can use something like this?
We've done some of that work, not as part of a trial, but certainly whenever you do an acute lengthening and there's bone defect left behind, the problem always seems to be limitation of autograft. That's the issue that is a huge clinical problem for us. And the solutions that we have are actually very expensive and poorly tested. So I think the science that we're doing, the studies that we're doing with the mesenchymal stem cell applications are actually some of the better science that has been done on this specific problem. So, yes.
So with that, and knowing how much work there is to do, I do just want to point out, thank you for those questions. And it is exactly this kind of science that will give more options in the clinical trials. And Diana, you've already benefited from one of the clinical trials and the use of mesenchymal stem cells, but we need to make it so that there are more options for more patients, for more indications, and that's what this is about. So I want to thank Kent, I want to thank Mark, I want to thank Diana for a very inspiring spotlight, and look forward to much progress to be made on repairing bones and using stem cells to improve lives. Thank you very much.